Bismillahirrahmanirrahim. So, Bismillahirrahmanirrahim. Excellency, Coordinator General of YC Comstech, honorable speakers, honorable participants, a good afternoon to you all and good morning according to your time. Inspired by the word of Nelson Mandela, education is the most powerful weapon which you can use to change the world. With this quote, it is my great pleasure to welcome you all to this webinar entitled Molecular Metabolomic Tools for Veterinary Disease Detection under the One Health approach, including policymakers, researchers, academicians, students, and experts from YC member states and the other region. Without further delay, let me invite to you all of you. Join me for the official opening of this webinar, beginning with recitation of Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Jazakallah khairan. Due to His Excellency busy schedule, he will join with us soon. Now I am pleased to invite Professor Dr. Marzia Rahman from the Department of Microbiology and Hygienic, Faculty of Veterinary Science at Bangladesh Agriculture University. She is a distinguished scholar in microbiology, antimicrobial resistance, food safety, and veterinary vaccine development. She holds a PhD in biomedical science from King Pong National University, South Korea, and has authored 82 scientific publications reflecting her strong research impact and leadership in animal health sciences. Professor Dr. Rahman will present on molecular and metabolomic tools for veterinary disease detection under the One Health approach. Ma'am, the stage is yours.
Thank you very much for giving the opportunity to present. Can you hear, please?
Yes, ma'am, I can. Maybe you are connected with the two devices. Could you please leave the one device and join with us? Only single device, that would be more suitable for us.
Can you hear me?
Yes, we can hear, but your voice is too low.
Too low in my computer.
Now it's okay, ma'am. Now it's okay.
Okay.
Yes. Now you can start your.
Bismillahirrahmanirrahim. Honorable organizer, uh, sorry, at first I am sharing.
Yes, Shyam. Pen.
Thank you very much for giving me opportunity to present the topics. Bismillahirrahmanirrahim. Honorable organizer, dear scientist and attendee, Assalamu alaikum. Welcome to my presentation. At first, I want to express my deep mourn. As you know, our former Prime Minister, Begum Khaleda Zia, has died today. Actually, today is very saddened day for us. However, I am going to present my topics. Today, the presentation topics is there is some problem. Molecular and metabolomic tools for veterinary disease detection under the One Health approach. Me, Dr. Marjia Rahman, Professor, Department of Microbiology and Hygiene, Bangladesh Agricultural University. At first, as you know, animal disease pose significant risk to human and human health. Human health mainly through the spread of antibiotic-resistant bacteria and other pathogens. Non-therapeutically, antibiotics are used in livestock as growth promoter that promotes the emergence of resistant bacteria that can be transmitted to human via food, water, and environmental interaction and cause us harmful effect. About 75% of the emerging human infectious diseases are directly or indirectly associated with animals, which highlighted the importance of health scientists working together as co-equal terms through a common platform, One Health. As we know, there are different kinds of pathogens can be transmitted from animal to human by using different transmission mechanisms, such as during wildlife hunting, HIV, Ebola, that can be transmitted from human to animal, from domestic animals, Hindra, Nipah, avian influenza can be transmitted, and while intensive wildlife farming, other COVID-19, rabies, influenza, and other organisms can be transmitted to human. In the same way, from live animal markets also, different kinds of infectious organisms can be transmitted from livestock to human. So, animal diseases remain a major constraint to livestock productivity and public health, necessitating accurate early diagnostic methods. Veterinary professionals hold considerable responsibility for the quick diagnosis, treatment, disease prevention, and nutrition of animals, working towards both enhanced animal welfare and human public health outcomes. Still now, traditional veterinary diagnostic techniques are useful, but fact that they cannot detect pathogens at early or subclinical stages. For that reason, To address these problems and welfare and economic issues, routine, robust, and early diagnostic tests are increasingly being developed and incorporated into practice. Thus, molecular and metabolomic tools are playing major role in the veterinary field with potential applications extending to animal reproduction, diagnosis, and treatment of animal diseases. So for that reason, the objectives of these topics are to gather knowledge on molecular and metabolomic tools for animal disease detection and biomarker discovery, to promote collaborative research and data sharing among veterinary institutes, laboratories, and universities within YC countries. What are the molecular tools used for veterinary disease diagnosis? Molecular methods are crucial in veterinary medicine for accurate and rapid disease diagnosis, including early detection before development of symptoms. The molecular methods enable precise identification of pathogens such as bacteria, virus, genetic disease analysis, and inform the development of new vaccines and therapies. These techniques also enhance surveillance and help to control outbreaks, improve epidemiology, and support international trade standards. Pathogens can be detected at low levels, identify specific strains, and are essential for controlling and understanding disease outbreaks. What are the molecular tools usually used for detection of veterinary diseases? The common method is the common tool of molecular methods are polymerase chain reaction. Different kinds of polymerase chain reactions are used for identification and diagnosis of disease, such as conventional PCR, multiplex PCR, nested PCR, real-time PCR, reverse transcription PCR. And additionally, microarrays, fluorescence in situ hybridization, DNA sequencing. These are the main tools, molecular tools, which are used for detection of veterinary diseases. How can we use PCR for veterinary disease diagnosis? It is used for detection of infectious disease, used in veterinary diagnostics for rapidly and accurately detecting pathogens like as bacteria, virus, fungi, and parasites. It is also used for diagnosis of disease at early stage, which is crucial for prevention of contagious diseases like contagious caprine pleuropneumonia in goats, mastitis, FMD, brucellosis, tuberculosis of cattle, avian influenza, mycoplasmosis of birds. PCR enables, it is also used for faster laboratory diagnosis than traditional methods. It allows for immediate disease control action, which is especially important for highly contagious disease. PCR can also be used to detect pathogen in environmental samples, which can indicate current or past infections within a facility. PCR is also used beyond infectious disease detection. It is used for genetic detection of genetic disorders in animals by analyzing specific gene sequence, which can be identified hereditary conditions like hip and elbow dysplasia or retinal atrophy, epilepsy in dogs, dwarfism, etc., of different animals. It is also used in conservation biology to assess genetic diversity within animal populations, which helps in breeding programs and managing endangered species by analyzing DNA from different sources like hair, feces. To investigate genetic basis of diseases and understand disease mechanisms by studying the DNA of affected animals. It also provides the genetic data, which is essential for the development of molecular-based vaccines such as DNA or subunit vaccines. How can we use microarray in veterinary field? What is microarray? A microarray consists of a solid surface to which biological molecules are arranged in a regular pattern. Microarrays can detect a variety of pathogens at once, including different subtypes of viruses, such as different subtypes of avian influenza, NDV, FMD, etc. It is also used for detection of genetic markers. It can identify specific genetic elements, including virulence factors, single nucleotide polymorphism, SNP, associated with disease susceptibility, and genes conferring antimicrobial or metal resistance to bacteria. Microarrays are used to analyze global changes in gene expression in infected hosts or tissues. It helps to understand the molecular mechanisms of a disease, identify novel diagnostic markers or therapeutic targets, and assess the host immune response to infection. It is also used for detection of novel pathogens that conserve the probes microarray and have the potentiality to detect previously uncharacterized or emerging pathogens. It is the speed and high throughput capabilities of microarrays are valuable for rapid response during disease outbreaks and for the detection of potential biological threats in environmental samples. It is used for development of novel diagnostic tests to develop and validate more focused and practical diagnostic tests for clinical use, different kinds of genetic markers, genetic variations, gene expression pattern, protein or autoantibody levels which are associated with disease detection are used to identify individuals at risk, classify diseases like cancer, diagnose infections, and predict prognosis. Fluorescence in situ hybridization, FISH, for detection of veterinary disease. FISH is a molecular cytogenetic technique that uses fluorescently labeled probes to bind to specific DNA sequences within cells or tissues. It is used for pathogen detection, such as for bacteria detection or bacterial disease detection, which are very difficult to culture in laboratory. That kind of bacteria detected by this method. It is also used for detection of virus directly in tissues by targeting viral RNA in paraffin-embedded tissue sections. That means by histopathology, immunohistochemistry. It is also used to identify viruses in tumors like Feliscatus papilloma virus in feline skin lesions. This method is used to identify fungal infections, especially pneumocystis carini in foals and pigs. This method is used for genetic disease and cancer detection in veterinary medicine to detect and to localize specific DNA or RNA sequence for disease diagnosis, including identifying intracellular pathogens and detecting genetic abnormalities such as chromosomal rearrangement and aneuploidy. It is used for diagnosis of cancer by identifying the genetic markers that are associated with cancer. What are the advantages of fish? It is it has high specificity, and it targets the DNA or RNA sequence, allowing for the precise identification of a particular microorganisms or genetic abnormality. Fish allows for the visualization of microorganisms within their natural tissue environment, providing valuable information about their location and distribution. It is used to provide results in a matter of hours, a significant advantage for diagnosing acute infections. It can be performed on various sample types, including tissue sections, cytological samples, and even in whole blood samples. Fish is frequently used to confirm the results from other tests, such as PCR and subculture, helping to provide a more definitive diagnosis. DNA sequencing for detection of veterinary diseases. Sequencing is the process of determining the order of nucleotides in a DNA molecules, which is crucial for understanding genetics, diagnosing disease, and more. Technologies like next generation sequencing and field portable nanopore sequencing enables the rapid identification of known and novel infectious agents, profiling of microbial communities, and tracing of disease spread and antimicrobial resistance genes. This allows for more precise, personalized, and rapid diagnostics for a wide range of veterinary diseases. Key application for disease diagnosis. It is used for infectious disease diagnosis caused by various kinds of virus, bacteria, parasites, or fungus. It is especially useful for detecting co-infections and novel or emerging pathogens that may be missed by other tests. It is used for profiling antimicrobial resistance by identifying genes that confer resistance to antibiotics, which is crucial for guiding treatment decisions and monitoring the spread of resistance. It allows for the tracing of how infectious diseases spread and for tracking the evolution of pathogens. This helps in understanding transmission dynamics and controlling outbreaks in animal populations. It is used for genetic predisposition by analyzing the animal's genome. Sequencing can identify genetic variations linked to inherited diseases, which helps in early diagnosis and treatment. Metabolomic and autoimmune diseases are detected by sequencing, by understanding the genetic and molecular underpinning of disease that helps in diagnosing and designing tailored treatments. Sequencing is used in oncology to diagnose and treat cancer by identifying specific genetic mutations in a patient's tumor. This allows for personalized medicine through targeted therapies, helps in cancer research by revealing how tumors evolve and become resistant to treatment, and can assess inherited cancer risk. Though it is very useful, but it has some limitations. Potential for false negative results due to inhibitors or degraded samples. The inability to confirm if an organism is the actual cause of disease, and difficulties in assessing treatment response because nucleic acid can persist after an organism dies. A positive result only shows the presence of the organism's nucleic acid, not necessarily the cause of the clinical signs. This is particularly relevant for organisms that can cause latent infection. These methods are generally more expensive, require specialized equipment and personnel, and are susceptible to false positive results from cross contamination. For that reason, as there are some uh, limitations in molecular methods, we need uh, some methods which are uh, available and can be useful for diagnosis of veterinary disease. In that case, metabolomics is the alternative or additional method for diagnosis of veterinary disease. What is metabolomics? We know metabolomics, it is the emerging field of omics science. It is a technique where small molecular weight molecule or metabolites of biological samples are analyzed to get a real-time snapshot of an organism's physiological state. It uses advanced analytical tools such as mass spectrometry and nuclear magnetic resonance to measure thousands of these molecules, revealing fingerprinting of health, disease, and environmental impact. Metabolites, what is metabolites? That we need to measure in metabolomics. Metabolites are the end products of cellular process, including nutrients, waste products, hormones, and signaling molecules that results from the breaking down of food, drugs, or tissues in body. We can determine or we can detect the metabolites by mass spectrometry, nuclear magnetic resonance spectroscopy, bioinformatics, and statistical analysis. What are the application of metabolomics in veterinary practice? It can be used for disease diagnosis and pathophysiology. Metabolomics is used to identify specific metabolic changes associated with various diseases, enabling earlier and more accurate diagnosis. It can be used for early detection of disease by analyzing different kinds of biomarkers, such as organic acid, amino acid in blood. This can be used for diagnosis of disease and understanding disease mechanisms. Other potential biomarkers such as lipid, nucleotide, hormones that which reflect the host response to infection, pathogen activity, and metabolic reprogramming. Metabolomics helps understand the disease mechanisms by comprehensively profiling metabolites in biological samples to identify metabolic dysregulation and biomarkers. By comparing the metabolites of healthy and diseased individuals using techniques like mass spectrometry and nuclear magnetic resonance, researchers can uncover how metabolic pathways are altered in diseased conditions such as cancer, diabetes, and cardiovascular disease. It can be used for disease diagnosis, such as for diagnosis of canine and feline disease. It can be used for diagnosis of diseases of livestock, such as different kinds of metabolic disorder, acidosis, hypercalcemia, fatty liver, etc. We can be diagnosed by metabolomics. Metabolomics is used to identify diagnostic markers for infectious diseases such as cryptosporidium infection in chicken. By analyzing an animal's metabolic response to diet, veterinarians and animal nutritionists can develop more targeted and personalized feeding plans. It helps in optimizing nutrient utilization, energy balance, and feed efficiency in livestock. This explored the effect of diet on specific conditions, such as using a high fiber diet for treating chronic diarrhea in dogs and optimizing pre-weaning nutrition in calves to maximize future productive outcomes. Metabolomics is a powerful tool in drug discovery and safety assessment. It helps to predict a drug's action, potential toxicity, and how it is metabolized within an animal's body. It can also be used to monitor drug residues in food producing animals to ensure food safety. Metabolomics helps in identifying metabolic biomarkers that are associated with desirable traits like disease resistance, growth performance, or product quality in breeding programs. It can be explored for early diagnosis and monitoring of cancer in both human and animals. Metabolomics detects cancer by analyzing the comprehensive profile of small molecular metabolites in biological samples like blood or urine to identify specific biomarkers that indicate the presence of a tumor. Mass spectrometry and NMR are used to quantify these metabolites, while spectroscopy methods can analyze tissues directly. It provides a deeper understanding of how various stimuli affect an animal's body system, which can inform practices that promote well-being. Contributes to advancements in the One Health approach, which connects animal and human health. What are the limitations? Complexity of data analysis, lack of standardized methods, difficulty in biomarker validation, and high inter-individual variability influenced by genetics and environment. It is challenging to find a single analytical method that can capture all metabolites. Loss of metabolites like glutathione may take place during tissue extraction. All metabolites cannot be detected. It is concluded that metabolomics will be a solution for animal problems in future. Thanks to all for your patience hearing.
Thank you, ma'am, for your insightful presentation. And would you like to give a few question answers? If you are free, then we can go ahead. Otherwise,
Actually, now I want to leave. I had a program just 3:30. As you know,
okay.
our former Prime Minister has died.
Yes,
So
yes, ma'am. I know that.
if you allow me, I want to leave. I have a whole day. We had a program,
Yes,
and
ma'am.
we're busy. If you kindly
Okay,
allow
ma'am.
me.
Thank you so much for your presentation. Basically, I am from Bangladesh. I know that the Bangladeshi situation right now.
Yeah.
So thank you so much again for your wonderful
Okay,
presentation.
thank you very much.
Okay.
Next time, inshallah, I will discuss about these matter topics.
Okay, ma'am. Thank you so much. Thank
Okay,
you.
thank you very
Thank
much.
you so much. And now I am honored to welcome our distinguished speaker, Dr. Wajed Amin Mirza, Assistant Professor at the Health Service Academy, Islamabad. He specializes in epidemiology and biostatistics and has extensive experience in public health research, infection control, and program coordination, having worked with various organizations. Professor Dr. Mirza holds a PhD in epidemiology and biostatistics and a DVM from the Jiangsu University, China. His research focuses on infection disease, infection prevention and control, and public health programs. His research is published in leading international and national level journals. Today he will deliver a speech on One Health, emerging and re-emerging disease. Sir, the floor is yours.
Oh, thank you so much. Hi everyone, my name is Dr. Wajid Amin Mirza and I'm from Health Services Academy, Islamabad, Pakistan. And I'm very thankful to you guys for giving me a chance, the organizers of the Comstech and all the scientists community and all the participants, the very vibrant and colorful audience. Thank you so much. So, One Health, today's topic is One Health approach to emerging and re-emerging disease. So One Health approach is not a simple topic, and I was given only 20, 30 minutes to explain it, and I'm not sure whether I can do it or not, but I'll try my best to do it. So the One Health approach, there are two kind of things. One is the One Health, and the other one is the public health approach. The World Health Organization has spent billions of dollars to combat the infection, and regretfully, he has not achieved his goal because he was just focusing on the infection prevention, not the prevention, but the control measures. So after years, like 10 or 20 years, here the WHO spent billions and they did not achieve their goals.
Professor, could you please turn on your screen in a big format?
Which format, sorry?
Could you please presenter view? You can go to the presenter view. We can see your presentation.
Okay. Presenter view. Okay, just give me 1 second, please.
Okay.
No, can you see my presentation or not?
Yes, you can see, but you can turn on in the big screen
Like this?
No. You can go to the home menu.
Mm-hmm.
Which one?
मुमताज को बोलो ना
Mumtaz?
उनको बताए ना कि कैसे करना
Yes. Wait.
Now you guys can see it or not?
Yes, we can see it. Yes. Okay,
Yeah,
thank you.
okay. Thank you. Thank you so much. So the goal of the public health is to ensure and to prevent the whole community, the whole population from the infection. And after achieving this, then a new concept, especially in Pakistan, emerges. That is the One Health approach. Because without adopting this One Health approach, we cannot control the infection in a community. So today's topic is the One Health approach to emerging and re-emerging diseases. So I will start with the One Health concept. The One Health is an approach to investigating infectious diseases, which acknowledges the humans, animals, plants, and the environmental as closely interlinked. So in the epidemiology, we also study the triad, the epidemiological triad that was included the host, the agent, and the environment. Without focusing on the host agent environment relationship, we cannot combat the infection. So the One Health concept is a little bit new concept in Pakistan, and it has like 6 pillars, which I will explain one by one. So in the mid 20th century, a veterinary surgeon in the United States called Galvin Schwab compared approaches to human and animal health and welfare and suggested the concept of one medicine. So it was not a new concept in the whole world, but in the Pakistan, it is a very, very new concept. So he highlighted the integrated cross-disciplinary perspective, which members of his profession could contribute to general medicine. So he also advocated involvement of social sciences and enhancing communication skills to improve working together with the community in efforts to control infectious diseases. So he highlighted the integrated cross-disciplinary perspectives which members of his profession could contribute to general medicine. So he also advocated involvement of social sciences and enhancing communication skills to improve working together with the community in efforts to control the infectious diseases. So there are normally more than 1,400 infectious diseases. And if you look at the data, there are 61 to 70% diseases which come from the animals. So without understanding the animal component, we cannot combat the diseases. We cannot combat the infectious in the humans, in the people. So the One Health approach integrates the human, animal, and the environmental components to combat the infection. So during the 21st century, this idea was developed further to encompass the health of the wider ecosystem, including that of the plants, wild animals, and geographical areas. The Manhattan Principles of Conservation, published in 2004, called for a more integrated view of the interactions between humans and animals. So if we go further, the 2002 and 2004 global outbreak of severe acute respiratory syndrome coronavirus shortly highlighted the risks posed to human by zoonosis. So the term One Health was first coined at around this time. This concept acknowledges that the health of humans, animals, and their behavior and their environment are closely interacted. And we cannot remove the infection only by focusing on the people or on the humans or on the population. The ecosystem translates as everything from the microorganisms living in our gut to the home we live in, the way we live our lives. Our ecosystem translates as everything from the microorganisms living in our gut to the home we live in, the way we live our lives. So although the term One Health has only been in general use since 2004, many of the linkages it explores have a long pedigree, with examples to be found in both Western Asian treaties dating back hundreds and even thousands of years. Because the humans and animals, they share the same environment. They've been living together for hundreds and thousands of years. So the zoonotic diseases which transmit from animals to humans, and these are not new. So these date back to even hundreds and thousands of years. So the United Nations Sustainable Development Goals are relevant to One Health as they include targets for health and well-being, clean water and sanitation, climate action, which are also known as environmental factors, as well as sustainability in the marine and terrestrial ecosystems. So recognizing the importance of the cross-disciplinary multinational working, for global organizations have now agreed to form the One Health quadripartite. So these are the World Health Organization, the World Organization for Animal Health, the UN Food and Agriculture Organization, and the UN Environment Program. They have identified 6 areas to focus on, namely, so these are the 6 pillars of the public health or One Health approach to combat the infection of emerging and re-emerging infectious diseases. So these include laboratory services, control of zoonotic diseases, neglected tropical diseases, antimicrobial resistance, food safety, and environmental health. So without working on these 6 pillars, we cannot combat the infection. We cannot control the zoonotic diseases, which contribute to the 70% of the diseases which transmit from animals to humans. So One Health approach to laboratory services. The first one is laboratory investigations are a key element for the diagnosis of infectious diseases. This is because their symptoms can often be non-specific, like in diarrhea, pyrexia, jaundice, the symptoms could be same. And full identification of the causal organism is important to determine treatment options. So many human pathogens are either acquired zoonotically or they have close relatives which affect other animals. So thus the One Health approach encourages medical and veterinary laboratory scientists to cooperate in the development of laboratory testing protocols without interlinked association between these departments like the environment, the veterinary side, the human side, and the antimicrobial resistance. When we take a holistic approach of these 4, 5, and 6 pillars, then we can combat the infectious diseases. Otherwise, if we focus only on the human side, that they do not know about the life cycle which the pathogenic organism has spent in the animals. And on the veterinary side, if we see only the veterinary side, the veterinarians don't know what is happening in the human body. So we need to take the holistic approach, and this intersector collaboration should be there to combat these infections, to combat these emerging and re-emerging diseases. So examples include, for example, many protozoan parasites of Leishmania species affect humans, as well as domestic and wild animals. So they are transmitted via sandflies of the phlebotomus or Lutzomia genera. So there are at least 53 species of Leishmania, of which 20 infect humans. So the WHO estimates that 1 billion people live in areas endemic for Leishmania infection. Prevalence studies have found infection rates of up to 25% in dogs, 10% in cats, and 50% or more in wild animals, such as rodents and in some geographical areas. So when it comes to the Leishmania diagnosis, there are two main problems for laboratory scientists to solve. The first is that therefore the Leishmania parasites found in clinical samples such as bone marrow and is the 5 micron intracellular MST GORD stage. This is difficult to detect with a microscope. The other is that there is a little morphological difference between many Leishmania species. Medical and veterinary parasitologists, along with entomologists, work together. We need to work together to develop and optimize the various tests of Leishmania. So although microscopy and serological tests are available, PCR protocols are the most useful as they provide acceptable sensitivity and specificity for diagnosis, surveillance, and research. This illustrates how the One Health approach can be used to share the experience and enhance capabilities in humans at veterinary medicines, as well as environmental research, because without the collaboration of all of these three departments, we cannot detect the Leishmania species very specifically. So it also helps in reporting notifiable diseases, and dead birds are sampled for avian influenza. So the aim should always be to perform the most appropriate test on the correct samples from the right patient and to report the results in the good time. So this idea of diagnostic stewardship fits well with the concept of the antimicrobial stewardship. So both are pertinent to One Health since they are approaches to using potentially finite resources carefully. So the second pillar is One Health approach to controlling of zoonotic diseases. So this is the main problem. More than 70% of infection is coming from animals to humans. So we need to focus on this area. So the zoonotic infections occur when humans interact directly or indirectly, because some people are keeping the pets, some are working on the farm animals. So this is the direct contact. And these animals also share the same environment with the humans. That is the indirect contact. So with either wild or domestic animals that harbor mutually transmittable diseases, so controlling therefore requires an appreciation of both the way in which these interactions occur and the factors responsible for maintaining the infections in those animals. So the One Health approach encourages us to operate the care and humility and always be open to the possibility of unintended consequences. For example, rabies was eliminated from larger parts of the northern Europe, at least partially, not fully, but partially, through the dissemination of baits laced with the anti-rabies vaccination. So rabies had previously been a major factor limiting fox populations, and therefore their populations increased. So foxes are the definitive host for the tapeworms, Echinococcus multiocularis, whose larval stage can infect humans, within which they form potentially fatal alveolar hydrated cysts. So the number of human infections with E. multiocularis have increased in several parts of the Europe, and one reason for this is undoubtedly the increase in the foxes populations, because they were not killing the foxes. So the number of foxes have increased, and they have encountered this problem. So from a human perspective, strategies to reduce the contact with the potential animal reservoirs or invertebrates, vectors of infection through education along with the treatment or prophylaxis programs should be there. So a One Health approach taking a wider view can be beneficial. We not only need to focus on the human health, but the animal health as well. Because when the animals are ill, when the animals are sick, they shed a lot of bacteria, they shed a lot of parasites. And as we share the same environment with the animals, so we can get the infections from the animals. So the importance of taking a global view of the control of zoonosis is illustrated by the outbreak of monkeypox in 2022. So the disease caused by Mpox virus, which is a member of the Poxobridae, endemic in West and Central Africa. So the virus was identified in monkeys held in the research laboratory in Copenhagen in 1958, hence the original name. So the first recorded human case occurred in what was then the Congo, now the Democratic Republic of the Congo, DRC, also known as the DRC, in 1970. The usual hosts for this virus are various species of the rodents which live in the west and central Africa. So, such as the African rope squirrel, this is also responsible. The infection is considered endemic in humans and countries where these rodents reside. So, reported case fatality rates range between 2% and 10%. So deaths usually occur in the very young, elderly, or immunocompromised, including the HIV positive patients as well. For example, between January and September 2020, there were 4,594 record cases of Mpox in DRC, with 171 deaths. So despite the public health measures in place to reduce the spread of COVID-19, this represented a 61% increase in cases compared with the 2018. So One Health to controlling neglected tropical disease. So the WHO recognizes 20 neglected tropical disease. These are also called the NTDs. These diseases are caused by a range of viruses, bacteria, fungi, and parasites, as well as snake bite is also included in the neglected tropical diseases. So they are mainly transmitted among poorer communities in low and middle income countries and are considered to markedly affect women and children more than the adult men. Examples of neglected tropical diseases include rabies and echinococcus, which are zoonoses where humans are infected accidentally. So bronchioliasis and schistosomiasis, which are caused by the parasites which complicated life cycles, intermediate invertebrate hosts, and lymphatic filariasis, which is transmitted to humans via mosquito vectors, and mycetoma caused by the exposure to infectious agents or irritants in the soil. So in the case of the mycobacterium infections, the organism is found in the environment, but the mode of transmission to the humans is still uncertain. We don't know the mode of transmission still. The conditions in which people are living, the quality of their food, drinking water, and housing all contribute to the risk of exposure to many morbidity from neglected tropical disease. So therefore, the One Health approach is important to better understanding the epidemiology of these diseases and defining the key to improved control of these infectious diseases and emerging and re-emerging diseases. Chagas disease can persist at a subclinical level for many years, and therefore people who travel or migrate from endemic regions may be unaware of their infectious status. And when they go to the next country, they can shed the infection, they can shed the disease over there. So in a woman of childbearing age, undetected infection is a particular problem. Since congenital transmission is possible, although the risk is low, about like 5%, so the antenatal screening is required in endemic areas before the migration. So the viral infection on the list of neglected tropical diseases include dengue, and which are both the positive sense single-stranded RNA viruses spread between humans by the bite of the Aedes species, so mosquitoes. So principally Aedes aegypti. So people infected with dengue can experience a range of symptoms from subclinical infections to a mild self-limiting illness involving pyrexia, myalgia, headache, nausea, or rash. Some people develop a positive viral type syndrome of extreme fatigue, muscle and joint pain, debilitations, and depression, which can last for months or even years. So the reported incidence of both these diseases have been increasing during the 21st century. This is partly explained by increasing surveillance and better diagnosis. So the laboratory testing for the viruses has made an important contribution to this, since it is hard to distinguish infections. So the PCR ideally multiplex assays are the optimal diagnostic approaches. However, these are expensive and not widely available in the endemic areas. Humans are the main reservoir host for both the dengue Although the surveys have detected evidence of infection in some wild animals, so the One Health approach to control is useful as it takes the environmental factors into account. So climate change is likely to be causing an increase in people's exposure to vector mosquitoes. Therefore, control of dengue infection should include public health measures to attend to the environment in endemic areas. There should be regular maintenance of water courses to remove the possible mosquito breeding sites, along with effective cleanup after floods. So better prediction of extreme weather events could be helpful. So this would allow plans to be put in place to move people to safety in time and to provide them with good shelter and food in their temporary living conditions. So then they should be less likely to return to their homes before suitable maintenance of the environment has been carried out. So the next component is One Health approach to combating antimicrobial resistance. So antimicrobial resistance is one of the most serious threats that will impact us in the coming years. Some scientists predict that by the end of 2050, the next pandemic could be the antimicrobial resistance, because you're giving the patients the antimicrobials, the antibiotics, and they are not functioning or working on them. So you can even die with simple infections. So the antimicrobial resistance is one of the most serious threats that will impact us in the coming years. So many pathogenic bacteria like Mycobacterium tuberculosis and Staphylococcus aureus are becoming increasingly resistant to all our currently available antibiotics. So this will impact on everything from routine operations to the re-emergence of the diseases in the countries from which they had been largely eliminated. So although the development of resistance is a natural phenomenon and to be expected to occur over time, the speed of development and spread of resistance among numerous pathogenesis is largely a consequence of the ways antimicrobial and antiparasitic drugs have been and continue to be misused. Although the development of resistance is a natural phenomenon to be expected to occur over time, the speed of development and the spread of resistance among numerous pathogenesis, largely a consequence of the way antimicrobial and antiparasitic drugs have been and continue to be misused. This is repeated. I'm sorry for that. This is included 2 times in the same slide. So everyone knows that the antibiotics have been oversubscribed for many years. In the United Kingdom, for example, it is now supposedly necessary to obtain a prescription from a medical doctor or other qualified practitioners to obtain an antibiotic. Although doctors are becoming less willing to prescribe antibiotics, they are often subjected to pressure from patients to prescribe them whether they are necessary or not. So unfortunately, even when antibiotics are needed, tests are not always undertaken to determine whether the antibiotics prescribed will be effective or not. It would be helpful if more accurate point of care tests that could be determined the species, subtypes, and also the drug sensitivity could be developed. A further problem in the United Kingdom is that it is possible to obtain was filling the simple questionnaire. So in some countries, there is an even laxer approach. Therefore, there is a need to either change the legislation or enforce existing legislation more stringently in the world. The combination of overprescription and incorrect consumption of antimicrobials results in massive amounts of them and their metabolic products being excreted and entering the sewage systems, which is also creating a problem. Additionally, many prescribed antibiotics that remain unused and end up being flushed down to the toilets and washed down the sinks. Within the sewage system, the antimicrobials interact with the numerous microbes and become dispersed into the wider ecosystems. Interestingly, the presence of microplastics, pollutants in the soil has been linked to the spread of antimicrobial resistance. This may be through bacteria possessing genes conferring antimicrobial resistance, forming biofilms around the microplastics, and thereby avoiding being washed through the soil. So the microplastics then become associated with crops. So another possibility by which the antimicrobial resistance bacteria can enter the food chain is by becoming attached to even smaller plastic particles. So those are called the particles that are absorbed by plant roots and then transported around the plants, and we consume those plants. So that's how the antimicrobial resistant bacteria reach the humans. Consequently, the food and water we consume and their direct and indirect effects on our health and that of other living organisms are not yet understood. So the widespread use of antibiotics means that many non-pathogenic microbes becomes exposed to them. So both within our bodies, such as in the gut microbiome, and in the wider environment. So consequently, numerous harmless microbes also develop antibiotic resistance genes. One health approach to ensure the food security and food safety are important for both humans and animals. The presence of toxins, pollutants, or pathogens in material destined for consumption is not always immediately apparent. So this highlights the importance of mechanisms for regular testing of foodstuff to ensure that they meet set standards and to ensure appropriate storage and transport. Where a specific risk has not been identified, it may be overlooked in quality measures. The One Health approach recognizes the effect of interaction with animals and environmental conditions on whether particular items present a risk to humans when eaten. For example, an outline in One Health to controlling neglected tropical disease section, Chagas disease is normally transmitted by blood-feeding bugs. So, however, in parts of the South America, there have been outbreaks of the infection caused by the consumption of fruit juices. So, the epidemiologists have investigated those. So, the investigations indicated that the drinks had been prepared from food contaminated with feces from the bugs infected with T. cruzi. So, the radioactive bugs had acquired the parasites from wild animals and subsequently defected on the fruits. And when we consume these kind of fruit or juices, then we get the disease. So the One Health approach for ensuring environmental health. So climate change is an increasingly significant factor in public health. It affects all communities and its consequences will undoubtedly become more serious in the coming decades. How it manifests varies between locations but can be broadly categorized as a prolonged rise in air and sea temperatures, accompanied by extreme weather events such as heat waves, floods, and cyclones and droughts. So these in turn destroy infrastructure, render people homeless, and reduce the production of the crops and livestock. Similarly, rises in sea temperatures and acidification in combination with rising sea levels is devastating fish stocks and causing the land erosion and flooding in the coastal regions. These problems are increasingly apparent in the higher income countries, but the consequences are far more serious for poor communities in the low and middle income countries, where they are leading to poverty, malnutrition, and economic migration. Some of the examples include climate change can influence the transmission of infectious disease in numerous ways, and the One Health approach is the only method is useful to understand how factors are connected. So extreme heat waves cause the migration, and the migrated people could have the so many diseases which can spread to the new areas as well. So One Health approach would also consider the environmental changes involved for invertebrates. Higher prevailing temperatures may affect their life cycles as well. So for example, provided there is sufficient moisture, hookworms larvae can withstand land temperatures of up to 40 degrees centigrade. And their developmental cycle can take place 3 times more quickly than usual. So this would enhance the opportunities for more human infectious mosquitoes could also go through their life cycle faster, increase their geographical range, and potentially overwinter in places currently considered to be too cold as global temperature increases. So the other big effect of climate change, which is possibly of greater significance for infectious diseases control, is the increased frequency and extent of very heavy rainfall leading to the flooding. So what is the conclusion of this One Health approach topic? The examples discussed here highlight the value of the One Health approach to investigating the cause, epidemiology, and control of the infectious diseases. So biomedical scientists and clinical scientists working in diagnosis laboratories, understanding the value of working in multidisciplinary healthcare terms, and review the complex clinical cases. There is a clear case for an analogous approach with One Health to address the complex multifactorial issues of infectious diseases requires inputs from a range of healthcare professionals, veterinary practitioners, social workers, and ecologists. The stated aim of the One Health quadripartite initiative include promoting a cross-sectoral approach and producing advice and policy guidelines for the governments, healthcare planners, and relevant national and international organizations. This must include funding to support the design of the multidisciplinary research programs and robust evaluation of their outcomes. So I just want to shed some light on the emerging and re-emerging infectious diseases. This could take like 4 or 5 minutes maximum. So infectious diseases have shaped human history, causing repeated global crises. So despite the advances in sanitation, antibiotics, and vaccines, infectious diseases remain a leading cause of global mortality. So optimism in the mid-20th century about defeating infectious diseases proved premature. So the recent decades have seen the emergence of the new pathogens and the resurgence of the previously controlled diseases. So they are re-emerging. So emerging infectious diseases and re-emerging infectious diseases pose sustainable threats to the public health, economic stability, and global security. Conceptual classification of emerging and re-emerging infectious diseases is that emerging infectious diseases are newly identified diseases in humans, known diseases with rapidly increasing incidence or geographic spread, often linked to the novel pathogens or mutant strains, like it happened in COVID-19. So the re-emerging infectious diseases are the previously controlled diseases that have resurged or resurfaced, often associated with drug resistance, weakened surveillance, or reduced vaccination coverage. Both categories continue to occur intermittently and challenge global health systems. So multifactorial determinants include human factors, population growth, urban overcrowding, poor sanitation, international travels as well. So microbial factors include the genetic mutations, the viruses and the bacteria, they are changing their genetic makeup. Adaptation, antimicrobial resistance. Environmental factors include ecological disruptions, climate variability, land use change, and the globalization include rapid cross-border movements of people, animals, and goods. So the public health gaps are the weak surveillance, delayed detection, and limited preparedness. So animal-human environmental interface, we also study this in the epidemiology. Without focusing on this triad, we cannot control the infection. So approximately 70 to 75% of the emerging infectious diseases originate from animal reservoirs. So zoonotic transmission is a primary pathway for disease emergence. Example includes HIV, SARS, MERS, COVID-19, avian and swine influenza, Ebola, and other viral hemorrhagic fevers, and increasing human-animal contact elevates spillover risk. So the early recognition of zoonotic threats is often delayed until outbreaks are well underway. So integrated approach to emerging and re-emerging infectious diseases, human health, animal health, we need to focus on these areas under the One Health umbrella. That is the human health, animal health, environmental health, promotes interdisciplinary collaboration across sectors, enhances early detection and surveillance, so coordinated outbreak response, evidence-based policy and prevention strategies, and essential framework for addressing the complex ecology of emerging and re-emerging diseases globally. So this was my today's presentation, and thank you so much for listening. If you guys have any questions, I'm here to answer all of them. I will try my best. Oh, thank you so much.
Thank you so much, Professor, for your wonderful presentations. And
Yeah.
it was informative and inspiring talk. Now we would like to go
thank you
for
so much.
a few question and answer session. So we have
Yes, please.
we have a few questions. One question from
Thank you so much.
one question from It's not showing. Israr Ghani, how can One Health frameworks be integrated into global policies to address infection disease, particularly in a resource-limited region with the diverse ecological and socio-economic challenges?
Yeah, this is like, as we can see from the topic, as we can see, the One Health approach is not like the small concept. It includes all the departments. It includes all the departments, the human side, the veterinary side, the environmental factors, the antimicrobial stewardship, and the ecology and everything. In these countries, we need to allocate more resources to such kind of problems. And like in the previously, like it happened, the doctors and the veterinarians and the specialists, the environmental specialists, they were not sitting together. They were not sharing the resources. They were not sharing the money. They were not sharing the budget. So we need to share these kind of resources, and we need to share these kind of knowledge. If something is happening in the human side, their conclusions, their abstract, their conclusions, their key findings should be shared with the veterinary side, and something is happening in the veterinary side, their conclusion should be shared with the human side. So intercollaboration, the holistic approach, and the you can say the resources, the money, these are working on these factors, we can contribute this problem, I think so, according to my view.
Okay. We have another question that is,
yeah, this is a multidisciplinary. Yes, please. Yes, please.
What is the importance of bioinformatics in One Health approach?
Yeah, bioinformatic analysis, as for the bioinformatic analysis is concerned, this is analysis we usually run on the genes, which I have performed. There could be many kind of bioinformatic analysis, which I have done on the genes. I have done on the dengue, because there are 4 strains of dengue, and the 4 strains of dengue, we need to translate their DNA. And after translating their DNA, we put that DNA samples, we put their DNA sequences, RNA sequences in a Mega11 software. And that Mega11 software compares our country's genomic data with all around the world. So with the bioinformatic analysis, we can see whether the strains of the dengue virus, which are circulating in our country, what is their origin. So through the bioinformatic analysis, we can see the origin of the dengue virus serotypes circulating in our country, whether they are coming from the India or any neighboring countries. So that's how we can use the bioinformatic analysis.
Okay, thank you, Professor. We have a one more question from the Mohammad Bilal.
Yes, please.
He asked, what are the reliable tests for the animal disease in field condition for the bacteria, viral and protozoan diseases?
Yeah, I have heard about this question. I was reading in this section, and this does not include in my field of expertise.
Okay.
So I'm not well sure what kind of tests are available in the veterinary side. So yeah.
Okay, thank you, Professor. Thank you so much for your
You're welcome.
beautiful presentation. I think we need more with you about One Health approach. And
yes, sure.
We extend our special gratitude to you for the informative discussions. Now I have.
Thank you so much.
Thank you. Now I have honor to invite His Excellency Professor Dr. Mohammad Iqbal Chaudhary, Coordinator General of YC Comstech, Mustafa Prize Laureate, and a distinguished national professor to deliver his vote of thanks. Sir, the floor is yours.
Thank you very much. Auzubillahi minash shaitanir rajeem. Bismillahir rahmanir rahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Thank you very much to distinguished speakers and a very important seminar which took place today on a very pertinent topic. I'd like to thank Dr. Marzia Rahman from the Department of Microbiology and Hygiene, Faculty of Veterinary Sciences, Bangladesh Agriculture University, which is a premier institution, And then Professor Dr. Amin Mirza. Dr. Amin
Thank you,
Mirza
sir.
is from the
Thank you, sir.
Faculty of Public Health, Epidemiology and Biostatistics and Health Services Academy, which is indeed a very good organization. So very happy to see both of you.
Thank you, sir.
Happens that both are young speakers, so
Yeah.
comstech is a very important occasion that we have two prominent young speakers speaking about a topic which is rarely understood actually. So, people talk about One Health, but as a concept is not very much clear to many of the health professionals, policymakers, even to the researchers also. And see, I was thinking that this concept has its own merit as a holistic approach because uh, we have been largely using our strategies as a reductionist approach. So you would agree with me that the hospitals are not healthcare facilities, but sick care facilities where we just take care of people when they get sick and there is no prevention available. And that is largely because we do not really understand a complex web of diseases which originate from animals to insect and then so many environmental factors which eventually lead to not only human getting sick from the viruses and pathogens of animals, but vice versa also. I remember Yuval Harari's book on sapiens, and in that book he talks about large mammals eliminated because human viruses have killed them. So many of these zoonotic diseases, say about 50-60%, experts would perhaps know the exact figure. Many of these diseases are of zoonotic origin, and it's very, very important that we understand the whole concept in its own merit with a broad-based understanding of what it means and then developing strategies to take care of it. We have been largely focused, even today, on physical health of human patients without taking care of understanding that it has many environmental and animal health and insect health and other aspect of it. So it was a very important concept which has been explained so nicely by two very competent speakers. And I really expect that both of them would actually spend more time. Rakib, that you have organized this, and we hope that we can have even a summer school organized on One Health with a greater understanding, more interaction, more discussions, so we understand this topic a bit better. I would like to thank you both, Dr. Marzia Rahman and Dr. Wajid Amin Mirza.
Yes, sir. Thank you, sir.
For excellent presentation, and Rakib and Akim for taking care of this very important seminar. I'm so happy that very large number of participants from different countries are attending this important event. So thank you on behalf of comstech. Assalamu alaikum.
Thank you so much, Excellency, and we sincerely appreciate the wisdom and inspiration in your words. With that, we officially conclude today's webinar. We hope this session has been insightful and productive, and we look forward to your continued engagement and future comstech activities. We are grateful to all the resource persons and online participants for their valuable contribution. Thank you so much.
Thank you. Thank you.
Assalamu alaikum.
Thank you very much.
Thank you.